Yes, absolutely. Sorry, Professor. Right. I mean, I can share my screen if that's possible. Uh, no, I'm not. Yeah. Oh, I think only hosts can share the screen at this moment. Yeah. Um, does it work? Okay. Um, okay. Yeah. So, so the presentation, so the title here is low-rank tensor-guided variational autoencoder, which we've introduced we've learned in class for incomplete multi-view information reconstruction. And due to time constraint, we can only do like clustering for now. And the inspiration for to pursue like a study in this subject is from this article, which is published November 2023, which is not too long ago. It is about predicting multiple conformations via sequence clustering and AlphaFold 2. So it talks about this main problem here, which is about predicting multiple conformational states of proteins, which is critical but an unresolved challenge in structural biology. And previous methods, including AlphaFold 2 that was introduced in 2021, it was excellent at predicting single protein structures, but it fails to accurately predict dynamic conformational ensembles, such as those seen in metamorphic proteins, which are essential for biological functions and disease understanding. And this paper introduces the idea of clustering sequence alignments by evolutionary similarity to capture a wide variety of structural conformations. And by leveraging these cluster inputs, AlphaFold 2 can identify multiple conformational stages with high confidence. And this demonstrates that sequence diversity encodes information about alternative protein states. And the proposed method, which we abbreviate as AF cluster here, it applies clustering algorithm dbScan, which stands for density-based spatial clustering of applications with noise, to multiple sequence alignments. It deconvolves evolutionary couplings and predicts alternative conformational stages, on top of AlphaFold 2. And this approach successfully identifies known and novel protein states, which is validated by experimental techniques like NMR spectroscopy And this advances our ability to map protein energy landscapes and functional dynamics. And you might ask, like, so with AlphaFold 3 here, why don't, does it really solve the problem? Well, AlphaFold 3 advances in atomic level modeling and diffusion-based techniques, which may improve sampling of alternative protein conformations. These features offer a potential edge in exploring conformational landscapes beyond the static predictions of AlphaFold 2. However, it still doesn't inherently address the challenge of reliably predicting multiple conformational stages or scoring them according to the Boltzmann distribution. And without direct integration of methods to deconvolve evolutionary signals or handle diverse data sets, like AlphaFold 3 can only partially solve the problem of predicting dynamic protein ensembles, which are essential for biological functions, but doesn't really inherently solve the problem. So yeah, the problems with the AF cluster here that I believe is, first of all, the use of DB-scan clustering algorithm. It presents several challenges. So first of all, it uses DB-scan clustering algorithm to process multiple sequence alignments by sequence similarity. So while this algorithm is widely used and I would say effectively, it's quite effective in terms of solving density-based clustering problems, it is not considered a highly advanced clustering technique. We have already developed like a much more sophisticated methods like spectral clustering or hierarchical clustering. So this algorithm might not address the complicated problems in the protein fold data. And the second one is the AF cluster relies solely on the multiple sequence alignment data, which raises questions about whether its like predictive power could be enhanced by incorporating different data. Let's say, and even like data of different modalities. So techniques like multi-kernel learning, it might provide a way to integrate additional information such as biochemical or structural data, and it might potentially capture a richer and more nuanced representation of protein dynamics. So yeah, the proposed method here, the variation also encoder based for like for the multi-view clustering stems from here as we try to incorporate more data into the analysis instead of just the multiple sequence alignment, because our belief is that is established on the fact that we think it provides insufficient information. And so, yeah, I don't think I need to reintroduce variational autoencoder, but the actual problem, the actual twist that I made here is on the prior distribution. I don't necessarily like it. It's usually a standard Gaussian. I think it's too loose because okay, so we enforce like we have this term the Kullback-Leibler divergence here to restrict the encoded posterior distribution. We don't want it to deviate too much from a standard Gaussian. But the thing is, I think there's a smarter way to actually solve this like restriction problem. We can, but at the same time, like I don't want to enforce a prior distribution that is too strict because that loses generality. I've seen like other research teams that use gumball softmax and like Gaussian priors, or they might use like a mixture of hierarchical, a mixture of Gaussians. Well, that might work on certain data sets, but it certainly does not solve all the problems. And I want the variation also encoder model to learn the multi-view data in more of a self-supervised manner. So these are the goals. And this is the proposed solution. So I mean, by view here, you can understand it in terms of the protein data is like structural data, biochemical data, and it just incorporate like data of different modalities to integrate the knowledge in more of a cohesive manner, and encode them into the latent space and calculate their their callback library, which I abbreviate as KL here. We calculate their similarity. Well, we notice that the KL divergence is by nature it's asymmetric and it's non-negative. So we use this term here to map it into a space where it is between 0 and 1, and it is and it's now become continuous, and it's now become symmetric. And also, I think this technique is also used in the loss function of contrastive learning, where we do not really want like one latent space to be like overfit to the other, or like and this kind of increases the robustness. But anyways, like this is, I think all the deep learning models are kind of black box, so I can't really give like a proof on this. Yeah, so we now construct a similarity matrix for the latent variables in each view, and then we and now the similarity matrices has now become symmetric, and we leverage the information of in other modalities if the data is currently missing in the other incomplete modalities, and yeah, and I'm gonna go fast here because I think this is quite long, and I can go long for an hour on the solution. But yeah, now we try to learn this using a transition probability tensor, and using a transition probability. First of all, we ensure that all the entries are at least non-negative, and they're all combined into one row-wise, so that this provides normalization. And this gives a groundness to the similarity that is learned by the callback library divergence, and now we stacked all the transition probability matrix into a tensor along mode 3, and then we perform tensor decomposition. So yeah, these are all the steps of tensor decomposition. We decompose that into an addition of commonality and specificity, and the tensor decomposition technique is used with alternating direction method of multipliers, and we iterate through these, and yeah, I guess this part is like it might be a little bit too long. Yeah, I do wish that I had more time to explain on this, but yeah, I'll just jump to the conclusion. Yeah, I'm running out of time. So, well, because of the lack of high quality labeled data sets in terms of the protein fold, I can only test the results on some very generally adopted data sets in computer vision. But I did find this data set called protein fold, which includes 12 views or let's say 12 modalities, which include composition, secondary, hydrophobicity, volume polarity, and many more. And the task is to do clustering on using all the information from the 12 modalities. And yeah, here are the results from the very common data sets from the computer vision, and the best results are bolded, and the second best results are underlined. So this means it really works. And yeah, here's the convergence. So I think I put it here, like the computation complexity of this model. It might seem a little bit intimidating, but actually it will always achieve like a convergence in less than 300 epochs. And the missing rate here is about like if I randomly drop like let's say 30% or 70% of the entire data, and I'm wondering like what would be the performance degradation, and it seems like it kind of maintains like a pretty good like robustness, even though like a significant amount of data are dropped. So yeah, here is the result on the protein fold. So the comparative methods are actually the multi-kernel learning models But as you can see, I didn't use multiple kernels in terms of my model, and the state of the art result on this is these two. Um, the ACC here means accuracy, and NMI you can understand like how you can understand it as some sort of like how good the model recognized the underlying clusters that this model learns, and so unfortunately it didn't beat the state-of-the-art model in terms of in terms of this very protein flow data set. But I'm also quite confused about the feature dimension here. It only has like 27 feature dimension, which I believe it doesn't really encapsulate all the data. And I do believe if the dimensionality is higher, my model will have higher performance. But yeah, it doesn't really. But even though I didn't use multiple kernels to understand like the data of different modalities, it still achieved like a pretty good, like a rather, I would say, acceptable performance. And it still shows like pretty good like robustness when like 10% of data are missing or 30% of the data is missing. So I believe it. So with further fine tuning or like some integration of like prior information, it will perform even better. So yeah, that's pretty much my presentation. Yeah, the work is now on arXiv. So if if you're interested in the details of the math, yeah, I'm really happy to discuss about it. Yeah. Thank you, Professor.
Hi everyone, this is Team 14. My name is Isaac, Beiyue, and Tao. Today we are going to present our final project. So before we jump into our project, we need some background information. So what is the promoter? Promoter is a specific region of DNA. They play an important role in indicating gene transcripting. Promoter contains specific DNA sequence that provide binding site for RNA and various transcription factors. Understanding and accurately identify the promoter region is necessary for the geometrics and molecular biology because it helps us comprehend gene regulation mechanism. And the next one we want to talk about is the DNA bird. DNA bird is a transformer-based deep learning model specifically pre-trained on DNA sequence. Inspired by the success of a bird in natural language processing, DNA bird adapted this architecture to handle DNA sequence by utilizing k-mer embedding. This model has shown excellent performance in various geometric tasks such as DNA sequence classification and transcription factor binding site prediction. Building upon the success of DNA-BERT2 introduced several enhancements to improve the performance further. One of the key innovations of the DNA-BERT2 is the using of bipair encoding. We can call it BPE. BPE helps reduce vocabulary size and capture more meaningful sequence pattern by combining frequently occurring substrate. So what is the problem? So the problem is that while DNA-BERT and DNA-BERT2 has made significantly long DNA sequence analysis, they have some limitation. Both models determine whether the promoter region exists within the entire DNA sequence. However, they do not have the capability to identify the exact position of promoter region within the sequence. In practical terms, this means that although this model can tell us if a promoter exists in the whole DNA sequence, they cannot tell us where it is located. This may have some trouble for other further research.
So now I'm going to talk about our approach and results. First of all, data preparation. We use ensemble database for DNA sequence and regulatory features. And when we extracted promoter positions from regulatory features file. We then formed 1,000 nucleotide long sequence containing promoter regions using sequence IO, which is a Python package. We choose 1,000 because in the original paper they used 10,000 nucleotides, but they only need to identify whether there's promoter in the sequence. So if we want to use 10,000, it will make our data very unbalanced. Then we created sequence label pairs. The sequence is the DNA sequence, and labels are integers indicating whether the nucleotide at this position is part of promoter regions. 0 means this nucleotide is not part of promoter region, and 1 means this nucleotide is part of the region. We use 2 species for training and testing. First of all, human. We use it for both training and testing with 80-20 training test splits. There are 24,213 instances in total. To test whether our model can generalize to other species, we also use mouse for testing only. There are 22,094 instances in total. We define and fine-tune a DNA-BERT-based model for token-wise promoter region positioning. First, we freeze all parameters of DNA-BERT2. Then we created a custom neural network model that adds new layers on top of the frozen DNA-BERT2 model, including multi-head attention layer and the classification layer that makes prediction at each token position. For the training, we use 200 epochs, and for training, we use cross-entropy loss. And for testing, we use accuracy. Here's the results. The fine-tuned DNA-BERT2 can reach the accuracy of 0.7836 in human testing set and 0.8218 in mouse testing set.
Based on the above illustration, and we also want to know that we have known that the DNA-BERT was training on a large-scale DNA data set, and it has many previous knowledge. And we also want to know that how the DNA-BERT will achieve its performance. So here we compare a naive BERT-based model. It will serve as a blank control group for comparison. Also, we want to know that if we employ a larger model, that is, a model will have more parameters, then how is its performance? So here we will also compare the performance of GPT-2-based model via LoRa. And for the specific experiments, we have aligned the experiment setting with the DNA-BERT. The first, we also use 200 apples to train model, and for the training, we also use the cross-entropy loss, and for testing, we also deploy the accuracy metrics. Here we have report the final results of the 2 methods. Yeah, and
in the end, we compare the results and provide possible explanations. We highlighted the best performing model in each species by orange. We can see GPT-2 performed the best in human test set, and DNABIRD2 performed the best in mouse test set. And naive bird performed the worst in most of the test sets, mainly because it doesn't contain the unique patterns specific to DNA sequences. However, GPT-2 outperforms DNABIRD2 in the human test set. We think that it's because its large size and diverse parameters and pre-training data. However, it lacks more generalizable biological patterns, which makes it underperform in the mouse test set. Here's the important significance and future developments. We showcase that existing model can be fine-tuned to not only detect the presence of promoter regions, but also pinpoint the position of those regions with decent accuracies. And our version of DNABIRD2 can generalize across species. These can be used to facilitate downstream applications. For example, DNA chisel. It is an application that can optimize the DNA sequence with customized constraints and optimization goals. By identifying promoter regions before running DNA chisel, we can focus on or avoid promoter regions that will have high impact on the expression level of the gene. In the long term, the potential use of this application can be gene therapy and synthetic biology. These are the references. Thank you. Any questions? 2 more minutes. No questions? Okay, yeah, let's welcome the next speaker.
Hello everyone. Today we're discussing our course project, and the problem that we're working on is how to learn better antibody sequences, or generate better antibody sequences from a language model given developability feedback. So the motivation here is, one, antibody design is an extremely important problem. Antibodies are proteins, as we discussed in class, proteins that are part of the immune system. And they have multiple functions. Those include killing pathogens by binding to and stimulating the immune cells, so it helps the body fight off disease. And it also can be used in cancer therapeutics, such as to bind to cancer cells and stimulate the immune system, or to bind to a cancer cell and then deliver a cytotoxic drug. One of the main issues with designing these antibodies synthetically is that there's a very large design space. So it's estimated that humans can make up to 1 quintillion unique antibodies. And in the body, these antibodies get rearranged until they find the right combination of amino acids to bind to the pathogen the body wants to attack. But when we're doing this synthetically, we need a very good way to predict whether these antibodies will bind to the target and how to narrow down that design space of 1 quintillion to a narrower subset that we can actually test in vitro and then in vivo to get a good antibody. So we're using computational methods such as language modeling to help us learn from these many antibody sequences that we have data on, and then select a narrow set of the promising antibody candidates. So the motivation here is that, one, we need multiple criteria needed for an antibody to work in humans. So a lot of models are trained on unsupervised data, so looking at trying to generate antibodies that kind of mimic the natural distribution of antibodies, or they might be going off of 1 or 2 design criteria, such as enhancing binding affinity. But in reality, there's a lot of different criteria needed for an antibody to work. For example, they need to be stable enough, they need to be developable so we can manufacture them, and we want lower toxicity and off-target effects in humans. Furthermore, many antibodies and drugs in general can be in silico on a computer and then in vitro, but then even though they might work in a petri dish and have high binding affinity, they then fail later in vivo, so like mouse studies. And even if they might work in a mouse, they then later fail in humans. So one important goal is being able to take in information. So for example, we have our 10,000 compounds maybe at the beginning that you're screening. 5 of them get to human studies. If we get information about those compounds that were used in human studies, how do we feed that information back into the model to update the model and enhance the model's probability of producing a successful drug? So one thing that we're working on is using IGLM. IGLM is a generative language model. It's trained on over 500 million antibody sequences, and it uses an unsupervised infilling training task. Basically, if you hide part of the antibody sequence, can the model generate the rest of it? These are conditioned on the chain, such as the heavy or light chain, and the species of the antibody, and then they provide a library with which you can choose candidate antibodies. However, it doesn't specifically optimize for tasks such as developability, solubility, aggregation, etc. So our goal is to allow the model to incorporate new information about additional properties such as developability to make better sequence predictions. And just some background on developability index. This is a proxy that we are using to show whether the antibody will do well in vivo or in patients later. Of course, this isn't a perfect proxy. We're doing this mainly due to resource limitations. We don't have a lot of patient data or the ability to do mouse studies in the time frame of this course. So we're using developability index to create our model. And developability index looks at a few different characteristics of antibodies, such as the thermal stability, so like the shelf life, aggregation propensity, so like are the antibodies going to bind to each other? Are they soluble enough to work in humans? And what is the risks of immunogenicity? So for example, how safe is it in humans? Looking at IGLM, we generated a few outputs from IGLM and looked at the developability index. And IGLM does not rank the antibodies in terms of developability index. So what we want to do is enhance IGLM so that it first outputs the developability indexes with higher or better developability at the beginning of its output. So we want to essentially fine-tune IGLM through reinforcement learning to get it to output higher developability index drugs.
Here are generally the background approach, which is reinforcement learning with human feedback, which is quite popular in current natural language processing studies. So as you know, ChatGPT-3 and GPT-4 are all like tuning based on human feedback to enhance their ability. So I think we want to adopt a quite similar approach. So the standard working pipeline for tuning language models are in 3 stages. So in the first stage, we will have supervised fine-tuning, which means we pre-train the language model or like any model on like a preset corpus. And secondly, we will train a reward model. So basically, we will have humans ranking the answers of the language model and train a reward model which can score the responses given by a language model. And finally, we will use reinforcement learning strategies for fine-tuning. And in our case, in step 1, we actually have got IGLM. So since IGLM is already pre-trained on the unsupervised antibody sequences, it can serve as a fine-tuning model in the stage 1. And then we need to consider training a reward model. So basically, in real-world standards, for say in our vision, we should have, for example, doctors or experimental settings where we can observe real features which can kind of show whether antibody sequence is good or not. So as Anna just said, currently in our experiments, we just use the developability score as our ranking, and we use like a predefined model which can generate developability index for any antibody sequence as our reward model. And why we choose this is generally because humans don't know which antibody sequence is better by reading it, and we probably need an analysis of which antibody sequence are the best. And the possible options for our choices are like performance in viral studies, efficacy in humans, or this developability index. And we are currently choosing it because it's just most simple for us to implement within a short period of time. And the final stage is the reinforcement learning, which we update the model from the real world. So we need to train a copy of the IGLM with proximal policy optimization, or IO, which is like memory efficient version of PPO. And we encourage the model to output the sequences that score highly within the real world model and penalize the responses that are too far from the original language model response. So generally this will be our overall framework. We have a pre-trained IGLM for antibody design, and we sample like a data set where like the model needs to generate the mask areas. So we have heavy human and antibody prefix as the condition to the model, and the IGLM actually needs to generate the masked region. And after we get the sample output, we will use the reward model, which is the developability index model, to generate a score for each of the sample outputs. And then we will use reinforcement learning algorithms like PPO or IO to optimize the model. So DPO, which is also like popularly used in language models, is not usable here because this is actually not preference-based data. We also take into other IO approaches into consideration. So for offline IO, we have behavior cloning and learning. However, behavior cloning does not work for this task because the sample size is actually not enough, and we do not have Q-learning currently very prevalent in large language models. So for policy gradient and reinforce, we find that the reward is actually too sparse and may lead to poor performance. And we have said that DPO is not useful due to the form of feedback. So we are currently trying PPO and IO.
And about the results, so to date we already demonstrated that the IGLM model does not order antibodies according to the developability. And so unfortunately the result is not ready at this moment yet, but we think that until the final report we can include them. But we do include some implementation details here. Like we already collected 10,000 IGLM generated antibodies, and we began policy gradient implementation. And we also implementing transformer reinforcement learning for IGLM. And yeah, because this is we don't have another chance for to report our progress in this lecture anymore. So we also discuss about the future plan about how reinforcement learning with human feedback will work for biology problems in real world. So we have some research questions like how to design the reward function that is, for example, for antibody design, what is the property that we care about the most? And also how to collect this data from real world. Like for reinforcement learning in LLMs, we can easily collect human preference data, while the effectiveness of a new drug is only shown in the later stage of the development, like when they are tested on mice or humans. So the challenge we need to face here is the reinforcement learning with low human feedback regime. So in summary, for this problem, we study the in silico design for antibody sequence that is really critical given the large design space of antibodies, and many models have already been created to generate antibody sequence like IgFo, IgRM, anti-BRT or Atlan. So, but all of them cannot solve the problem of the gap between how well antibodies work in silico, in vitro, in vivo, and in the real human bodies. So selecting antibodies for qualities likely to make them more successful in later stage, we improve development process in both saving time and effort. Yeah. So that's pretty much our presentation. We're happy to answer any questions that you have.
Yeah, I think we ran out of time, so let's move ahead to the next one. Thank you.
you could upload it to OneDrive.
Hello everyone, my name is Linju. And today we are going to talk about the project. It's a homework project, so it's predicting the binding affinity of RELs with a FYP protein and based on the GN model. So firstly, I will talk about what's RELs and what's YFP protein. So what's YFP protein? YFP protein is yellow Florence protein. So it's just a mutant of the Florence green Florence protein. GFP is green Florence protein. Why we want to take Florence protein? Because it has a special shape. Like Florence protein is have the we can from the picture it looks like that one, and it's a barrel, and the inside is it's from the top view, and it looks like the shape like this thing. So it could be interactive like with else and something. So and also it's very stable, so we can do a lot of mutations about on the Florence protein, and we can still keep their structure. So if the structure will blow up or change their shape. So yellow Florence protein and the green Florence protein are members of structurally homologous class of proteins that form a chromophore from a sequence of 3 amino acids. So it's very structurally stable. So we can try many different mutants without worrying about the structure integrity, and also the dynamic of the proteins do not contribute too much to their binding capacity. So this is the plot of which side you do the protein, and it will change to like the yellow Florence protein and the cyan Florence protein and the blue Florence protein. So next is what's RELs. RELs is just that specific these L's, and it's very the mass is pretty big, and it could be very special properties like high magnetization and the collective activities and these specific properties, and they also more usually in the conductivity designing. So and the last one is our data is MD simulation data. So we running the MD simulation system. So we set up the simulation systems like each the proteins we do the 110 the mutants as a single mutants. So our box set is 87 angstrom, and the concentration is 0.2. So each system they have 24 RELs, and for the chart. And so as a computer to keep our system natural charge, so it's we include this one, this else, it correlated, yeah. And the total atoms is about 60K. So each systems we run the simulation is about 1 nanosecond. So it's a pretty long trajectory. So that's when
So the inspiration for this project comes from a paper that was on the association of water to proteins based off of their charge of the surface. So this structure on the left is a fluorescent protein showing the charge with negative in red and positive in blue. And the findings from the paper of the association of water to a protein based on charge was that it wasn't just on the local average, it was more complex than that. Because there are, when you parameterize a structure for simulation, you give charges to all the atoms. So there can be charges from something that has an extra electron or is missing an electron. But then there's a sharing of electrons through covalent bonds. So some of them can be shared more towards one or the other. So you can have a local positive and a local negative. And even though it's averaged to be neutral, this can affect the ability of it to bind to the charged molecules they bind.
feature instruction like the to do the graph presentation. Like so we have the protein PDB file. So we extract the protein PDB from the trajectory, and then we present it to be the graph. That we calculate the distance between two atoms is less than 5 angstrom, and it can be have the each. And then the node is atoms in the protein, so it's around 3,700. So the input feature is just because we don't want to put a lot of features first, so we just using the 3D coordinates and the charge for each atoms. And the labels, we calculate the average time of the having major ions spending within 6 angstrom of any atoms of this residue. So we could totally can get the 240 dimension for each protein. So we are going to it's a multiple label task. And for the data set argumentation, so we do the protein rotation. So we want to generate more data. So the protein is rotated around X, Y, Z, so randomly, and so we do 100 times for per mutant. So it totally have 11K argumentative samples. So it's inspired by AlphaFold 3. So this will improve the performance by forcing the model to not memorize where each atom is, and this artificially give us more data for the model to training on.
So for the overall architecture, we use GCN with, we do 2 cycles, and each cycle has a fully connected layer for the node, and then a layer for the neighboring nodes, and then that's the neighboring node features on average, and we do 2 cycles for that. And then after that, we have a fully connected layers going from these features down to the final output, which is the mean residence time for the ions. And then we're using mean square error loss. And then so far, I reused very few parameters, and that wasn't working, and fewer layers, and then adding it in improves it, but then eventually a lot of the outputs and the weights would decay to 0. Changing the optimizer to one without weight decay and using slower learning rate will fix that, but it's still not really in the range that we want, because it's outputting around 2 to 6 for all of them, and then the real values actually go from 0 to maybe 120 or more. And yeah, something that we're thinking of doing, adding in, is putting in the solvent accessibility values to make it easier for the model to learn, because the residues on the interior of the protein can't bind to the ions, and that could just initially fix some stuff. And then maybe using edge features for distances between atoms, maybe even saying if they're covalently bonded, and then trying residual connections or different weight initializations. We just used uniform and chiming uniform. And uniform was just a little bit better to start, because we started between -1 and 1 instead of the chiming uniform was like standardized to be smaller.
Yeah, maybe we'll also consider to fine-tuning existing model to our task, because now we just training a new, very initially simple DNA model on our data. Yeah, is there any questions?
Yeah, we're at time. Okay, thank you. Real environment.
is our presentation on our knowledge distillation of protein language models. In particular, we distilled ESM2 to BERT.
So unlike other groups which are focused on like specific model development issues for like specific problem, we're more interested into how do we distill the existing knowledge from large protein models to a smaller model.
So like our key motivation for this problem is that protein language models are expensive to train and perform inference, and this is mostly due to the scale law, as larger language models can learn the protein language better. And we just want to, our goal is to get a cheaper model to learn and distill the knowledge from those larger models into a smaller one, which helps to improve accessibility of models. One challenge is that, like, at least for the time limit, models like AlphaFold 2 are pretty complicated, so we landed on distilling ESM2 to BERT, as ESM2 is a pretty similar model to BERT. So the model we used in particular had 3 billion parameters, but it can go up to 15 billion, while our BERT model is only 110 million parameters. And challenge was that there's really no well-known student models of protein language models. There are models for large language models that have done knowledge distillation, but it's pretty novel for protein language models.
And in the beginning, we have a look at OpenFOAD, which is a open source implementation of the AlphaFold 2. It turns out that the internal architecture was so large that we were not able to replicate a smaller model. For distillation, so we pivot into ESM instead of trying to replicate on. Previous work, those knowledge distillation are mostly for large language models in the NLP, and there's an open source tool from Microsoft that they have existing pipelines for knowledge distillation from large language models to smaller image models. And they also have a pipeline for knowledge distillation during the pre-training for other large language models, but all those models are based on are distilled based on the prompts, and they heavily relies on prompt to do the largest distillation. And also there's early work of DistillBird, which I think they cut on the center bird size from like 110 million to like 59 or 57. With faster inference speed, but with 97% of the original performance of the center bird. And here we're trying to distill knowledge from ESM2 into a center bird, which is just 110 million parameter.
So this is a diagram of how we trained it. So we used the data from a Uniref 50, and we limited the max sequence length to 512. So we put the input we use the ESM2 tokenizer, and then we feed that into both of the models, and then we use KL divergence to compute a logits loss with ESM2, and then we we still use the separate mask language model loss. We use cross entropy to compare to the expected labeled sequence. And then we calculate a total loss by just combining both of the losses into the total loss using alpha, which is a hyperparameter, which we can change the ratio to based on training.
And because there was no previous pipelines with how they're actually doing this for training models, we were thinking of firstly pre-training that the amino acid sequence, so you will have a relatively closer largest output to the ESN, and it turns out the logit loss was it grows even higher when we are training in the during the pre-training process. So instead we just go straight with alpha equals 0.5. Puts equal weight on both the amino acid sequence masking as well as the largest loss, which is the KO divergence between the largest output of the ESN2 and our own model. And it turns out they converge really fast. And there are some preliminary results. We're still running the rest because the evaluation tests there are totally 9 evaluation tests for just on the ESN sequence. That those 3 are extracted from the protein BERT, which is a BERT model specialized trained on protein sequences. And it turns out that with only 10 million parameters compared to the 3 billion parameters, we were able to achieve some substantially accuracy compared to ESN secondary structure predictions, but our model's performance on remote homology detection, especially the flow resistance intensity prediction, were really bad. Because I'll look into the data, and it turns out that it was a sequence. The label is just a numerical value, and I think the BERT model itself does not really encode the numerical value really well. So that turns out to not doing well during a knowledge distillation.
So due to the limitations of our time, we probably could have done a lot more training. So I think further training and hyperparameter improvement would probably help improve our results by just a little bit. And we've noticed that the logit loss could be kind of unstable, and the KL divergence grows pretty large. So I think balancing and normalizing that better could help improve performance. And
we're also thinking of further distilling not just the largest output, but also the intermediate layers, as that could create more meaningful knowledge distillation for our smaller model.
And we could also do a training pipeline, like instead of splitting it into pre-training, just MLM and then switching to both, there could be a smooth transition where the alpha changes over time to use more of the logit loss to learn more from the ESM2 model. In the future, it could be okay.
Thank you. Let's go to the next team because we have 3 more teams. All right, thank you.
Okay, hello everyone. We're group 4, and next time, Mohsen and he worked on the temporal dynamic and predictive modeling of bacterial gene expression. First, I give you an intuition about the data set that we worked on, which was the gene expression pattern of E. coli through cell cycle. After single-cell RNA-seq, when we measure the correlation between genes when sorted through the chromosome, we saw three types of highly correlated genes. The first one is the neighbor gene. The second highly correlated genes was the genes equidistant from the origin of the replication. And the third type of the correlation that we saw is the origin and terminus proximal genes. Then when we mapped the single-cell RNA-seq to a 2D UMAP, we saw a circular pattern that showed the trajectory of cell cycle of individual cells. When we look at the gene expression sorted through cell angle and the position of the genes, we see replication fork shape inside our heat map that kind of shows the direction of replication. At the end, the data set that we have is the time series of all the individual genes in the E. coli through the cell cycle. Then in the paper, they use OD model to describe the behavior of the gene expression. They say that there is a steady state that depends on the transcription rate and degradation rate of the RNA, and then after the gene replication, we have a 2x increase in the gene mRNA copy number inside the cell, and after the cell division, it gets back to the steady state. Using this model and two other types of the model, we could apply this model and fit these models to the individual single cell. In the two other versions, we assume an activation model and a repression model as well, that through the activation window, we have a transient overshoot of the 2x in the gene expression, and in the repression window, we see a transient repression in the gene expression. And through this, we could describe about 90% of the behavior of genes. However, through this model, we don't have any idea about the gene-regulated network and gene-gene correlation. So we decided to look at and inferring the gene-regulated network out of the out of the single-cell RNA-seq data that we have. And for doing this, we did some preliminary steps. The first one is using the neural OD to use a single function to describe the behavior of all the genes inside the cell. First, I give you a briefing overview of the neural OD. So when we have a time series data, we can use an RNA encoder and encode it to the latent space, and then encode it to the latent distribution. And out of this distribution, we can sample from it and set it as the initial value for a neural ODE. And then we solve the ODE that the function is like multi-layer perceptron, or it could be whatever. And again, we can use a decoder to go from the latent space to the data space and compute loss, and fine-tuning the parameters of the function to have to predict it accurately. Also, we could do it for the future time steps and extrapolate for future time points as well. So I applied neural OD on this problem. What I did is, what we did was using a temporal data encoder like GRU and give it like the first 20 time points of the gene expression pattern. And also I have a baseline data encoder that takes the start position of the gene, end position of the gene, and DNA strand that the gene located on it. And then we combine the result of these two encoder and DNA combiner. It's a one linear layer, and then set it as an initial value for the OD solver. And then we try to predict the unseen time points and use the decoder to see the result. And the function that I used for the OD was a 3-layer NLP with ELU activation function. The results that I got so far was not that much interesting, and the model could not predict the gene expression in the unseen time points pretty well. So our plan was to fine-tune the architecture of the model and also try some data augmentation. Maybe it could help us to get a better results.
While neural OD was one option or one method we were exploring, we also thought we could try our luck with like RNNs and LSTMs for the same gene expression level prediction task. So here I'm just motivating the input we give at each time step to our RNN and LSTMs. So one thing is that, let's say the expression level of a specific gene, it tends to be correlated or dependent on the expression level of genes around expression level of genes that are its neighbors. So you see in the first one, like expression level of gene G tends to depend more on genes from A to F and genes H to N. Similar for the expression level of, let's say, another gene, gene S. But there's one other feature here that's important, is that the position on the genome also matters. So you see there's a different mapping for the first, we have a mapping F, a function F for the first, when we try to predict expression level of gene G, while we have another mapping like function G for the expression level of gene S. So this is our intuition. So the two features that we care about, that we pass into our LSTM or and RNN, was the expression level of neighboring genes and also the position in the genome. So we also tried a many-to-many RNN and LSTM approach, where, as I motivated in the last slide, the input feature vector takes in the gene expression levels at each time step of its neighboring genes, and also the position in the genome. So our expectation was that our output, the output of this many-to-many sequential model was gene expression of our interest gene. So we trained this with mean squared error loss, and we also, the reason we use a sequential model is that these hidden states, they're going to help, let's say, a future gene expression level of interest gene here be dependent on the past expression levels of its neighboring genes. So in doing this after training, unfortunately our loss did not converge well, so our results were somewhat all over the place. And so we were able to identify some reasons as to why. One being the fact that our data was definitely limited to train such a somewhat sophisticated model. So the main reason is that our data was limited to learn the impact of our final feature here, the position on the genome, because that has a huge impact. If I go back to the previous slide, we have quite a different mapping from our neighboring genes depending on our interest gene. So for every gene, we need quite a lot of data for this model to output quite good results. So one such direction forward that we would want to do is, can we come up with a model where we can make it aware of this functional dependence of the position on the gene, as opposed to expecting it to learn from somewhat limited data. And the other one is, we expect that there are so this, I want to introduce this before. So this is just a gene expression level correlation matrix. So some genes, they tend to have similar correlation to their neighbors. So we were thinking maybe those genes that have similar correlation with their neighbors could have a similar functional mapping, which would mean we could cluster these interest genes and then try to train a sophisticated model, which would mean we have a lot more data and the intricacies could be learned. So these are two things that two takeaways from our initial experimentation. And our hope is once we find a model that predicts this interest genes expression level accurately, we can actually try to infer the gene dependencies.
I'll just go, I know we're running over, I'll go quickly. The whole idea that Hassan mentioned, gene regulatory networks are a big field, and this, you can think of them as they're dynamic heterogeneous graphs. So each gene is basically can be a regulator, can be a suppressor, any type of different role, two different types. So we're trying to really isolate how do we get a handle on the roles of each node, of each gene, and the links. So another approach we're looking at is looking at causal time series models. For reasons here, they seem to be natural models for helping us determine link prediction with respect to gene regulatory networks.